Assalamu alaikum wa rahmatullahi wa barakatuh. Peace be upon you all. And welcome to this new episode of Defining Truth. Is evolution true or false course? Today, this is the live stream about the antibiotic resistance. And why is this important? It is because in many cases, when you are talking with an evolutionist, or looking at a science explainer program, you will find in many cases that evolutionists would use the argument from antibiotic resistance to support the claim of evolution. They will tell you evolution is a fact, and when you tell them that it is not a fact because it's not observable, they will tell you, but it is indeed observable, and we observe it in the lab in the form of bacterial evolution. So you start in the lab with bacteria that used not to resist antibiotics, and you end up with bacteria that are resisting antibiotics, and this is evolution. So they would use the argument from antibiotic resistance as supporting evidence for evolution. So, in this episode today, we are going to discuss whether this is true or false. I have posted during the last week an episode about antibiotic resistance from our On Evolution series. And I hope that you have watched it. But even if you have not watched it, you can watch it after this episode to know more about what we are talking about, and you can still participate. Also, I would encourage everybody to participate to the Is Evolution True or False course by going to the website and registering there. So you can also send me questions by email prior to the live streams on Saturday, according to the topic that we are actually discussing. So this live stream is about discussing this episode of antibiotic resistance from the On Evolution series. It's episode 11, and this is the 7th live stream. So after this live stream, if you have not seen this, see it. Make sure you have also registered to the course, and you will find the link to the course in the description. So let me first start by giving you a short presentation to summarize the topic, and then we can proceed with your live questions. So I'll be sharing my presentation now. Okay, so antibiotic resistance is not evolution, is the topic of our live stream today. So first, what is antibiotic resistance? So antibiotics are those drugs that we humans have invented during the last century. And it was Alexander Fleming who was very well known to have discovered penicillin, which was a great lifesaver for millions upon millions, maybe hundreds of millions of humans, especially in World War II. And of course, it was not only him, but also two other important scientists, and all three took a Nobel Prize for this great discovery. And essentially, antibiotics are really a discovery rather than an invention. And we will know why they are actually a discovery. So first, what is antibiotic resistance? Antibiotic resistance happens when germs, like, for example, bacteria and fungi, develop the ability to defeat the drugs designed to kill them, and those drugs are called antibiotics. So you start by using a drug like penicillin. In the beginning, it just completely makes you get rid of the pathogenic bacteria that's causing you disease. But after some time, people start noticing that penicillin is not that much effective as it used to be, because guess what? The bacteria that penicillin used to kill does not die from penicillin anymore. And this definition I'm actually getting it from, this is the CDC, from the CDC website, and you have the links while I'm going through the presentation. Now, who invented, if we have discovered antibiotics, who actually invented antibiotics? Actually, nobody invented antibiotics. Antibiotics were there all the time. Actually, what Alexander Fleming did was a very smart observation that while working in the lab with bacteria, a little piece of black mold was in one of his petri dishes, and he noticed that the bacteria around the black mold did not grow. So he thought maybe the mold was producing a substance that is actually killing the bacteria, and he purified that substance, and he called it penicillin, and this was it. He discovered that this fungus was actually naturally producing penicillin. And what we have discovered even more, that actually one of the greatest producers of antibiotics that kill bacteria is bacteria itself. So for hundreds of millions and actually billions of years, bacteria and fungi have been producing antibiotics in this chemical warfare among each other. So you, for example, you have a bacteria that lives in the soil, and this bacteria is not as fastly growing or as aggressive as other bacteria that live in its surroundings. So what that bacteria would do maybe is that it would produce antibiotics that will kill off the other competing bacteria in its surroundings so that it can flourish and reproduce with less competition around it. And it's really amazing that these things are there in nature. And guess what? If one kind of bacteria is actually producing antibiotics, it means that that specific bacteria will not die of the antibiotics it is producing itself. It is, like the snake that has venom, but the snake venom itself, the snake doesn't seem to be worrying about the venom in its own mouth. And hence, bacteria and other microorganisms have been working with antibiotics for billions of years before we even had the slightest idea about what they are. So this is one very important thing to keep in mind in this discussion as we go forward. Now, what are the claims of the typical evolutionary speaker? He will tell you, look, he'll give you something like this, and what we're seeing in front of us is a big a container that has a fluid, and in this fluid, you will have a gradient of antibiotics. So the outer rim has a very low concentration of antibiotics, and the more you move in, there is a higher concentration of the antibiotic, and you start with bacteria at the sides, and then you notice as days and days go, the bacteria start proliferating and growing. You can see those spots where certain bacteria start branching from them, and as time goes, bacteria can grow more and more into the much higher concentration of antibiotics. And then he'll tell you, hey, look, this is at the right and the left. This was bacteria that was couldn't resist antibiotics, and as time goes, the bacteria develops this function. And guess why it develops this function? It's evolution. It's evolution in action. And they will claim that this is because the bacteria mutate, and due to this mutation, natural selection will pick the mutations that are most beneficial into fighting the antibiotic. And this process proceeds on and on until the bacteria can completely coexist with the antibiotics. And they will tell you, hey, look, this is evolution in a petri dish, and this is how evolution works. And so evolution is a fact and is an observable fact. And this is the danger of this argument from antibiotic resistance. And to understand why this is actually not an argument at all, we need to review a few things together. So, for example, I'm giving you here on this presentation two links. I'm going also to add the links to the description of the PowerPoint. I'm going actually to even put this presentation on the Evolution course website. So the first one is a link to the BBC. I'm going to watch to see this on the website with you after I finish the presentation. And on this website, it's saying, okay, and evolution is evidence. And the other one is actually a research paper from Science Daily about antibiotic resistance and why it is evidence or an example for evolution. So nobody can say that science explainers and TV shows and debaters about evolution are not putting this claim forward. They are, and even research papers is doing this. Scientists are doing this. But what is evolution that we are concerned about? Evolution is this claim that life has started from a very simple origin, single unicellular organism, and then kept branching and branching. So there is common descent, universal common descent with modification, okay? And the modifications are created essentially by random variations, and those random variations in the genetic material are acted upon by natural selection to pick the variations that make sense or may have benefit. And then life diversifies and gets more complicated, and the tree of life that we always hear about, which is like a picture like I'm putting in the presentation, takes place. But is this a process of just selection? No, it is also a process of continuous injection and addition of information. There is information, new information all the way, because the composition of a lion and an elephant is obviously much more information rich and much more complex than a single unicellular organism. So the first last common and universal common ancestor was a very simple cell, and then we have bacteria and archaea, and then we have eukaryotes, and then we have multicellular organisms, and then we have all the diversity of animals and plants that we see around us, up to the human being. So with each of those stages of evolution, there is addition of information. We are looking at the claim of evolution that life came from zero to hero through a process of common descent with modification by natural selection acting on random mutations, and that this process is in continuous generation of information and addition of information. So now let's look if antibiotic resistance is anything like this. But before we do that, let's look at a very important thing at play here, which is natural selection itself. Let's consider natural selection alone. What would natural selection do? You have a herd of rabbits, for example, and those rabbits are attacked by a tiger or a lion or a cheetah or something. So the rabbits that run fast are the rabbits that are going to survive, but the rabbits that are lazy or slow or diseased are the rabbits that are going to be eaten. And the same would apply for gazelles or antelopes, etc. Regardless which animal is being attacked by the predator, there is a process of survival of the fittest, which will cause that the animals who have the best features, the best body build, the best of this and that, are the ones that are going to survive, and hence are the ones that are going to have offsprings and reproduce. And this process of survival of the fittest is a process of selecting the best from the existing. So the animals that run, the rabbits that run faster are the rabbits that are going to survive. But you will not find rabbits that will develop wings. Okay, regardless the selective pressure that you are going to put on the rabbits, they are not going to develop wings because a tiger is running after them, because the necessary genetic code for wings does not exist in the genetics of the rabbit. So under this selective pressure, okay, the rabbit that has the best features of rabbits will survive under the threat of predation. If we have a new threat, for example, that we have an ice age, and then we have rabbits that have a better layer of fat or have thicker fur, those are the ones that are going to survive. And then you will find a predominance of that specific feature in the rabbits that will live in that area that was struck by a change in weather and is suffering from an ice age-like environment. But you are not going to find that under this selective pressure, you will find rabbits, for example, that are growing coats, for example, or that are growing a mechanism that makes them radiate heat, for example, or any other science fictional kind of thing, or they are going to have a new kind of leather on top of their coat or something, okay? Because this feature does not exist in rabbits, all right? So selection is the trait that gives advantage is preserved and multiplied, and selection by definition is to select from what exists. And selection alone cannot make a rabbit become anything else but a rabbit. And it cannot also make a bacteria become anything else other than a bacteria. So why is this very important? Because life on Earth, according to the fossil record, has stayed in this stage of unicellular organisms floating around in oceans for so long a period from the history of life. So if life started on planet Earth 3.8 billion years ago, we know that the Cambrian explosion is 540 billion years ago. So we have something like 3.3 billion years out of 3.8 billion years where you only have a very primitive life, okay? Maybe like 2.5 billion years or something where you only have things like bacteria. And if we know, if somebody can prove to us that bacteria, through a process of common descent with modification by natural selection acting upon random mutation, bacteria can become something other than bacteria, because the claim of the theory is that bacteria and archaea later became eukaryotes, and eukaryotes moved from unicellularity to multicellularity, and then you have the radiation of the different kinds of animals and plants, all of this happening by random mutations acted upon by natural selection. So the importance of this argument to the pop culture of evolution is to tell you that yes, bacteria can do it. Yes, bacteria can innovate completely new functions when there is pressure. But is this what is happening? So let's look at exactly what's happening. The reality of the case is that there are two, or we can even consider them three, mechanisms where antibiotic resistance surfaces to our level of observation. The first thing is something called plasmids. So if you look at on the screen, you will find this is a schematic of a bacteria, and in the middle here you will find the blue thing is the genetic material of the bacteria, and the red rings are called plasmids. And those plasmids are rings of they have rings of DNA inside of them, like you see at the bottom. And this ring will have, for example, an antibiotic resistance gene. And the bacteria will not carry all kinds of plasmids, are like wild cards. So this population of bacteria has this amount of plasmids, and that other one has other plasmids. And the interesting thing is that bacteria can actually exchange plasmids. So one bacteria can come, you can see that on evolution 11, some bacteria can come in proximity to other one, and like a pipe will extend from one to the other, and a process called conjugation will transfer one plasmid from this one to that one. Even sometimes they are different types of bacteria. So one bacteria will give to another bacteria a plasmid that it has, which means that the bacteria now, the one that has taken from the donor, has the ability to transcribe or to decode this new genetic material and produce something. But let's notice something very important, that the plasmid already existed. And one of the very interesting things that we have discovered later about antibiotic resistance is that you will find that we invent a new drug, we start killing harmful bacteria with it, and then another bacteria, maybe another completely different type of bacteria, or the same type of bacteria, has one plasmid that has genetic material that can produce the antidote to that drug. And this plasmid will now be distributed to the bacterial population, and the bacteria will start producing the antibiotic using this plasmid. And those plasmids have proven to be a very important thing, to the extent that now humans are manipulating plasmids and even doing some genetic engineering to plasmids to use them actually for our benefit. So this is one primary mechanism of antibiotic resistance. And under this mechanism, there is no mutation whatsoever to begin with. And hence, the whole premise of the bacteria was dying from antibiotics. So under the pressure, the bacteria developed a way to fight the antibiotics is completely false. Is this not known to the scientific community? Of course it is known to the scientific community. Why are they not mentioning it when they are doing their science explainer programs? You need to ask them. For example, I'm giving you here one plasmid that has a genetic material called NDM1 that produces an enzyme when transcribed, and then this enzyme produces a material, produces a protein that will almost destroy a complete category, action of a complete category of antibiotics. Let me give you an example. For example, how does penicillin work? There is a cell membrane for the bacteria, and the cell membrane has a material called peptidoglycan, and this material absorbs water. And there is a cell membrane, and the water would go from outside into the inside by osmosis through this material. And this material, peptidoglycan, gives the cell membrane support so that the water being absorbed inside will not cause the bacteria to burst. So this peptidoglycan is reinforced by an enzyme, and this enzyme has a very long name, so they called it the penicillin binding protein. And what this does is that it creates a matrix, okay? Puts those building blocks of the peptidoglycans together so that it forms this mesh around the cell that gives it support. So what penicillin will do is that it will go to the active site of this enzyme and blocks this enzyme from creating this matrix. This weakens the cell membrane, and hence water will be absorbed by the bacteria and it will burst. And bacteria can then disable the action of penicillin by actually attacking the piece of the penicillin that goes into the penicillin binding protein and disabling it. And actually, what NDM1 does is that it produces this enzyme that will disable not only penicillin, but almost every other antibiotic that uses the same mechanism like penicillin. And this plasmid was there on Earth in bacteria for billions of years, nothing new. Except that when you kill off so many bacteria that does not have it, the ones that have it will remain, and it will start exchanging it with the rest, and then you will find that bacteria is not killed off anymore with your medicine. What else? Sometimes even bacteria do not use plasmids. They can just switch gears. So this is, for example, on the screen here, another type of penicillin binding protein, which is the one that creates the green matrix you see up there. And there is two types. One type of them is susceptible to penicillin to bind to it, and another type called penicillin binding protein 2A is produced by another variant of the same bacteria, and this one, penicillin, does not bind to it. So when you kill too many of the one that has the penicillin binding protein, the standard one, let's say from our perspective, the others that have the penicillin binding protein 2A will prevail, and you will find that penicillin is not anymore being lethal to the bacteria. And this is, for example, something that we find in a very dangerous kind of bacteria called MRSA, for example, which stands for methicillin resistant Staphylococcus aureus. Okay. So this kind of thing, you will find evolutionary biologists telling you, well, yes, This is because a mutation happened to the penicillin binding protein, making it become penicillin binding protein 2A, and then this mutation is selected by natural selection. Sorry, what is your evidence that this is a random mutation? What is your evidence that this was not there all the time? And once again, there are sometimes also other processes. For example, bacteria have those interesting mechanisms where there are something that we can consider like pumps. And those pumps will pump out bad material or bad drugs from the inside of the cell and throw it out. And what happens is that when you start using antibiotics against bacteria, it was experimentally proven that those bacteria will react by creating more of those pumps in their cytoplasm, to the extent that they will expel more and more of the antibiotics. And there are other, sometimes they also claim that, for example, that this bacteria is coming to bind to that specific protein or that specific organelle in the bacteria. And then a mutation causes this protein to be disabled or dysfunctional. And then the antibiotic will not find the proper protein to bind to. And then this disabled bacteria will then become not susceptible to that antibiotic. And then when the threat of the antibiotic goes away, due to plasmid exchange and horizontal and vertical gene transfer of the bacteria, that function will be repaired or be reproduced, but the bacterial population would have survived the threat. Under all of those, the lesson learned here is like this. Bacteria is developing antibiotic resistance not because bacteria has an evolutionary brain that is causing completely random events to happen that are then filtered by a genius natural selection. The matter of the fact is that antibiotics have been on Earth for billions of years before humans even existed. Antibiotics were part of a biochemical warfare between bacteria and each other, and between bacteria and fungi for billions of years. Bacteria already have in their machinery the necessary mechanisms to fight antibiotics that are historically produced by their own comrade bacteria, whether by having plasmids that have the antidotes, whether by pumping out the antibiotics, or even using protein structures that are not compatible with the antibiotic so that the antibiotic does not hurt them, or even, if you would like to count this as a fourth thing, enduring disabilities to the extent that the disability will make the bacteria an inefficient binder to that antibiotic, and hence the antibiotic will not harm him. And in this case, I'm giving the funny example of a policeman, for example, who is going after a thief to handcuff him, and then when he reaches the thief, he finds that the thief has no hands, and in this case, the handcuffs will not work. So you will need to maybe use a rope. But if all the antibiotic that you have with you is a handcuff, you cannot catch that specific thief. You need a new type of antibiotic, which is, for example, in this case, a rope. So that was a fast presentation of what antibiotic resistance is. And let's now go to listening to some of your questions on the live chat and seeing if we can have some interactive discussion here. So Mark Ward says, Assalamu alaikum wa rahmatullah. Assalamu alaikum, Mark, and welcome to the channel. So let's see if we have some questions about the antibiotic resistance. You can put the questions right on the live chat, and if there are no questions, we might also conclude the episode. All right, so seems we don't have so many questions today, unlike the last time we had plenty of questions. So if this is the case, I would like to remind you all to subscribe to the website and to the course so that you can think more about the episode at hand or the session at hand and send questions beforehand. And you can also find organized articles about the subject, and you can even answer a quiz about each session to test your knowledge. And just one lesson learned out of this: even if they show you something with your own eyes and tell you this is evolution in action, we have to be skeptical about what is shown, because the metaphysical and the logical and the mental part of what evolution claims just simply does not make sense. It does not make sense that completely random events will generate novel information that will fit in a pre existing system to the extent that a single unicellular organism, like looks like bacteria, will develop over no matter how much amount of time to become a human being or a tiger or a cat or a mango tree. Having said that, thank you very much for joining this live stream. And let's have your questions pre-sent by email for the next episode. And let's put our extra to link some more videos to you. And see you tomorrow, where we will be having, inshallah, a nice interview with a medical doctor discussing the wonders of God's creation in the human skeleton. So till then, see you again soon, inshallah. And assalamu alaikum wa rahmatullahi wa barakatuh.